Okay. So good evening everyone and welcome to the week 8 live class session on the course Fundamentals of Protein Chemistry. Today I will be taking on metalloproteins and some enzymes associated with it. Okay, so yeah. So this is a course which is being taken by Professor Shagata Dasgupta of IIT Kharagpur and Srijan Sen, and today I will be taking the week 8 live class session. Okay, so I'm sharing my slides with you all. So I hope I am visible to you all. Yeah. Right then. Okay. Okay. So till the last class we talked about the proteins, enzymes, like that. So in today's class, our main focus will be on metalloproteins and motor proteins, and there are a few important enzymes whose cofactors are some metals and all. Let us see how we go through it. So first, I would be going with the brief lecture, and then I will be solving a few question and answers. Okay. So the way it goes is like what metalloenzymes actually are. So urease, for example, you have urease as an enzyme that catalyzes the hydrolysis of urea into carbon dioxide and ammonia, and this reaction occurs as NH2 whole to CO plus H2O, where you get carbon dioxide and ammonia. So aconitase is one such metalloenzyme, where you see in its core there is a metal ion that acts as the cofactor for the functioning of aconitase to break urease to break urea into carbon dioxide and ammonia. So you have the urease enzyme, right? So there are known biochemical functions of selected trace elements that are known to account for a sizable fraction of cellular metals in marine or microorganisms, like manganese, where oxygen evolving complex of PS2 superoxide dismutase. Then you have iron, where you are aware of the aconitase, ferredoxin, FES centers. You have the cytochromes, nitrate reductase, nitrite reductase, then cobalt for carbonic anhydrase, nickel for urease. For plastocyanin, ferrous oxide, you have the copper, zinc for carbonic anhydrase. Also, you have the role of cadmium. Now, the roles of metals in biology is different from that of chemistry, as understood by Lippard and Berg. So, metalloproteins function differently, like you have the dioxygen transport, where you have the hemoglobin, myoglobin family, hemocyanins, hemethrins. Then you have the electron transfer, you have the nitrogen fixation. For structural roles, there are some zinc fingers. Now, metalloenzyme functions are metalloproteins that perform a catalytic function. Hydrolytic enzymes, you have the carbonic anhydrases. Two electron redox enzymes, you have the nitrate reductase, oxidation of hydrocarbons by P450. Then multi-electron pair redox enzymes, the cytochrome C, PS2, nitrogenase. Then rearrangements, there is vitamin B12. Now, there is an important role of metalloenzymes in photosynthesis. From the diagram, you can see this is a photosynthesis, that the apparatus of oxygenic phototropes, where it illustrates that proteins that contain the iron with the potential number of electrons at different step is superimposed. Like so, you see over here you have LHC, that is the light harvesting complex. Then it goes into the cytochrome B. Then there are different photosystems that are active, where you have the 680 and the 700, and everywhere the role of metals or metalloenzymes are very, very prominent in every photosystem, be it photosystem 2 or photosystem 1. Now, metalloenzymes in photosynthesis, from here you can see that the iron content of proteins or protein complexes that are majorly involved in photosynthesis has been put up in the table. Like for photosystem 2, you have 2 to 3 iron atoms per protein complex. For photosystem 1, there are 12. Then, for example, let us take ferredoxin, you have 2. Then ascorbate peroxidase, you have 1, and then you have NADPH-PQ oxidoreductase, that varies from 8 to 18 iron atoms per protein complex. Now, this is about the role of metalloenzymes in the carbon fixation. So what is important over here, you see that in wherever there are different biological roles present, like the carbon fixation, you see that there is a conversion of bicarbonate to carbon dioxide, and there are certain enzymes that need zinc, cadmium, cobalt to activate them, or they are the cofactors that are important for the processing of the enzymes to carry out the entire um, biological process. Like you see over here, this is a nitrogen utilization pathway. You see the cell membrane over here, and then the entire cycle is on, and here you have the role of ureas, like nickel present over here, then molybdenum, uh, iron. So these are the things. You have copper over here. So every metal have different biological roles, and the biological processes are connected through the different enzymes, and these enzymes have different cofactors, like you have learned about cofactors. So these cofactors are metals. So uh, enzymes that require metals as their cofactors can also be termed as metalloenzymes. So this is another process where you see there is conversion of nitrate to nitrate conversion. Then you have ammonia. So this is basically production of ammonia, and you see there is copper, then you have iron, molybdenum, then what else is there? So in nitrification you need iron, then for ammonium oxidation there is iron and copper. For NO conversion there is like iron and copper again, and for the nitrogen fixation there is iron and molybdenum. So this is just a brief understanding about metalloproteins. Okay, now we will talk a little bit about motor proteins. Like there is nothing very much to tell over here as well, but still there are some things that you need to know. So motor proteins, from the name you can understand that it helps the cells in mobility, or the protein in moving. So it helps you to move. So motors are the things that helps you move. So actin based motility, like you have the myosins, you have the tubular based motility where you have the kinesins and the dyneins. So the role of actin-based systems is that it helps in cell crawling, like you have the lymphocytes, growth cone extension, then muscle-like contraction of ovarian follicles, mammary gland ducts, etc. You have the muscle contractions, then distribution of vesicles, intermediate filaments and organelles within the cytoplasm. All of these things needs to move from one place to another, or the cells are very active, and therefore the mobility of these cells and the proteins involved in it are very important, the function. So this is the role of an actin based system. Now you see over here the head is both actin binding and the ATP binding. The purple light chain has a regulatory role over here, and myosin 2 is the muscle myosin over here. So you have the two heavy chains that are intertwined between them or coiled around each other, and you have the lighter chains that are shown in purple. So all of these, this is actin based motility, the myosins, the role of the myosins. So the study of the movement of myosin against the actin filaments is an in vitro motility assay. So preparation of actin cables pointing in the same direction is used cells from a giant alga that uses these cables for moving of the chloroplasts around. Now there are some open algal cells and yellow fluorescent beads coated with myosin, and then add ATP and take time-lapse photography, and that will give you an idea about the motility of the alga, and from there it will fluoresce at a different region, and that's a very nice in vitro assay to test that. Now this is a cartoon of myosin coated bead actin preparation. So you have a bead over here, you see it's so you've seen this. This is a myosin and it's intertwined, and this is the actin, so it moves and just uh, interacts with the actin. So the speed of the movement, that is myosin coated beads moved unidirectionally and movement was dependent on ATP. The speed of beads coated with muscle myosin is around 5 micron per second, which is the same speed as the contraction of sarcomeres in muscle. Now, different myosins move at different speeds. Smooth muscle myosin moves at 1 micron per second. Okay, so these are the important points that you need to keep in mind. Now, for the study of movement due to a single myosin molecule, we can use a setup in which a focused laser beams creates optical traps, and these optical traps can hold the small objects. The force is controlled by adjusting the intensity of the laser beam. So you have the laser beam over here, and then there are beads attached, and then there is a glass slide. So it passes through the beads into the glass slide. Now you see over here there is actin filaments is held in a optical trap via one or two attached beads. Now myosin concentration are kept low so that only one myosin contacts with the actin filaments, and that will help you to actually do that particular assay. So over here you see the actins interacting with the myosin. Now ATP is also kept low so that only one ATP binds to each myosin head. So if I go back to the place where I was speaking about this, so you see the myosin 1 has an actin binding domain, that is also the ATP binding region. There are one light chains. For myosin 2, you see that it is like two heavy chains are coiled around each other. The sizes are different. So this is the head portion. The purple region is the neck portion. So the lighter chains are more complicated as you go from myosin 1 to myosin 5. And these two heavy chains tails coil around each other. Now the results show that the myosin pulls on the actin filament in a stepwise or ratchet-like fashion. So a single ATP molecule is hydrolyzed, resulting in a power stroke, and the displacement is around 10 nanomolar. And the force generated can also be determined, and it is around 3 to 7 pico newtons. When you plot force versus time, you can actually calculate that. Okay. So till now, do you have any questions? Any questions till this much? See, this is a lot of there is nothing much to actually understand this, like a lot, but you have to just keep a few things in mind, like about the structure, the function of the metal enzymes, what are the metals involved probably in this and that. So, if you have any questions, you can ask me. Yeah, no, till this part. So, okay, then let us proceed to the next part. That is the distribution of a vesicle along the actin network is polarized. So you have a pointed end, then the actin filament, and the barbed end. So this is starting from the pointed end, that is mostly you can consider towards the left side, or the starting point is the pointed end, and then you have the barbed end that is towards the end. So the myosin one head is the one that interacts, and then you can see over here there is a membrane vesicle around it. So myosin 5 is the link between actin, which serves as a rigid skeletal element, and the intermediate filament, which is being delivered to another part of the cell. So this is the intermediate filament fragment. You have the light chains over here, and the coiling, the intertwinkling. The head portion is actually interacting with the actin filament in this particular manner. So on the other hand, so this is about the actin based motility. Now the tubulin based motility is the role of this particular system is the exoplasmic transport, positioning of organelles within the cell, mitotic spindle, then they have roles in cilia and flagella. Now, exoplasmic transport is the, like, you have the anterograde, that is from from the cell body towards the synapse, that is towards the positive end of the tubule, driven by kinesins. Retrograde, it is mostly driven by the dyneins, that is from the synapse towards the cell body. Now, fast works for cargo carriers in vesicles, that is 500 to 400 per day, and sometimes for individual proteins it might be slow, but the net rate is less than 10 per day, but apparently this reflects a stop and go aspect of the process. Now the functions of exoplasmic transport is that it delivers proteins, mitochondria, vesicles to the synapses, then it removes recycled proteins and organelles to the cell body for destruction by the lysozymes. So these are the very, very important points of motor proteins, of proteins that are driven by the actins or dyneins. Okay, so the mobility, the cell motility is very, very critical over here, and it carries intracellular chemical messages from the synapse to the cell body and delivers neuron specific viruses, like you have the herpes virus, the pox virus. These are the some of the very dangerous viruses, fatal viruses, from peripheral sensory nerve endings to the cell bodies and the central nervous system. So you see, this is the structure of a dyne. You see there is some nice flower like things over here. It has, so this is the head domain. Okay, so for the kinesin, you see this is the head domain. You have the coil. So over for the dyneins, you do not have any coiled coils, but kinesins have a coiled coil. Then the light and intermediate chains are present over here, although it seems like a flower or a different shape, but it's are actually chains, I mean light and intermediate chains. So the name of these chains is based basically by the presence of the different types of amino acids that are present over here and the complexity of its coiling. Okay, and then what you see over here, so kinesin has a head domain, coiled coil, and a light chain, whereas for dyneins you do not have the coiled coil, but it has the head domain and the light along with an extra intermediate chain. So kinesin is thought to have evolved from myosin. So you can see that there is a very similarity in the structure as that of myosin, that is maintained for kinesin as well. So it has been thought that through the natural evolution or something like that, kinesins have evolved through myosin. So yes, there is nothing much to tell. We will be talking through when we probably discuss with the questions, because there is a lot of things that you need to remember. So even if I put it up in the slide, it becomes quite complicated even, because there are different portions that can be touched, might be missed out, but yeah. So okay, so any questions on the lecture part? Because next I would be starting with the question and answers. Yeah, actually, there is not very much from this chapter, or it's not even that complicated. You just need to keep in mind a few important things, mostly about the actin motility, the time motility, and something like that. So yeah, keeping those things, the structures and all, and that things will actually do it. Okay, so let us start with the question and answers, and then in the middle, if you have a question or something, we can address that. So you people can give me an answer, and then probably I will tell you. So the first question says, which of the following is or are true? Myosins are actin-based motor systems. Dyneins are actin-based motor systems. Myosins are tubulin-based motor systems. Kinesins are tubulin-based motor systems. You guys please try, and then I will tell you. So then let us try to figure it out. What is the answer to this? So uh, we know that there is only one actin-based motor system protein, that is the myosins. So myosins are actin-based motor systems. So myosin 1, 2, 5, these are actin-based motor systems. And dyneins and kinesins are actually tubulin-based motor systems. Okay, so dyneins are not actin-based motor systems. Kinesins are tubulin-based motor systems. So 1 and 4 is correct over here. That is option C. So option C is the correct one over here, because myosins are actin-based and kinesins are tubulin-based. Next one. Which of the following is or are true? Dyneins show retrograde transport. Kinesins show retrograde transport. Kinesins help to move the chromosome along the microtubule, and the ATP bound state of kinesin weakly binds to the microtubule. Which of the following is or are true? So like if I go back to this particular slide where I was speaking about retrograde and anterograde, so from here you can understand, like retrograde you have driven by dyneins, anterograde it is driven by the kinesins. So dyneins have retrograde transport, that is right. Kinesins have anterograde transport. Kinesins help to move the chromosome along the microtubule, that is right. And ATP bound kinesin strongly binds with the microtubule. So option A is correct over here. Okay, the next one. So this is a little bit might be tricky, but you can actually note down if you have to. So from the image, you have to identify the parts that are marked. So you have to mark what is X, then these circular like structures, that is Y, and then comes Z. So Z is basically this portion, this black, little bit of black. So what is that portion? So which one is what? Oh, and also you have to actually identify the image. So the image is like, is it an axonomal kinesin or is it an axonomal dyne? Any answer to this? No? Okay. So X, so whatever X you are actually seeing, this is basically radial spokes. So X is radial spokes. Then if you look at Y, you see that there is like two. So for every microtubule, you see that there are two microtubules. So it's a doublet tubule. And depending on whether it is a dynein or kinin, you can understand that. So I do not have a kinesin structure, but so these are dynein arms. Okay, and the image is of an axonomal dynein. X represents your radial spokes, Y is the doublet microtubule, and Z is the dynein arm. So it is like axonomal dynein. The next one is myosins are in a high energy state when the head is bent at 90 degrees. Myosins are in a high energy state when the head is bent at 45 degrees. Actins are in low energy state when the head is bent at 45 degrees, or actins are in a low energy state when the head is bent at 90 degrees. Any answer to this question? So the correct one over here is myosins are in a high energy state when the head is bent at an angle of 90 degrees. And myosins are in a low energy state when the head is bent at 45 degrees. But we cannot tell that for actins. So that is why the only correct option over here is A. That is, myosins are in high energy state when the head is bent at 90 degrees. So which of the following is false? ATP synthesis takes place when stroma has high proton concentration relative to thylakoid lumen. Gamma acts as an endogenous inhibitor of ATP synthase. ATP synthesis takes place when gamma shaft rotates clockwise, and ATP synthesis takes place at beta subunit. So those who have studied about this, they will be able to tell, like, so which of the thing is actually false over here. So this has nothing to do with metalloproteins, it's more of a ATP synthesis pathway. So the correct option over here, or rather the false one over here, is like option B. So gamma acts as an endogenous inhibitor of ATP synthesis. That is the wrong over here. But other options that you see, like ATP synthesis takes place when stroma has higher proton concentration relative to thylakoid lumen. That is right. Then ATP synthesis takes place when the gamma shaft rotates clockwise. That is also true. And even ATP synthesis takes place at the beta subunit. That is also true over here. So gamma acts as the endogenous inhibitor of ATP synthase is not the correct one. Okay. The image shown above is the active site of cupridoxine. What is the geometry of this structure? So you have to tell me the geometry of this particular active site of cupridoxine. Is it tetrahedral, trigonal, pyramidal, trigonal, planar, or pyramidal? So what is the structure of this? So after a long time I have got one answer. Okay. So tetrahedral. Okay. So tetrahedral is like every angle is I don't remember like how much will that be? It's okay. It's like 4.28 something like that, or 8 or something. But you see over here you have a planar structure. So it's not a planar structure, it's actually a triangle. So it means that there are three sides. So it's a trigonal. Either it has to be pyramidal or planar. So pyramidal is like you do not have anything on the top or the bottom, but a pyramidal will have on the top, and then you have the lone pairs over here. So it's a trigonal pyramidal structure. So B is the correct option over here. Tetrahedral is somewhat like you have a middle atom and then the other three distributed across the three arms at an angle of 104.28, like that. But over here this is like 120, 120 and 120, and this will be like the angle between the top atom and thing over here is like 90 degrees. So 120, 120, 120, and on top and bottom the axial positions have 90 degrees, whereas the equatorial positions have 120. Which of the following is false regarding hemocyanin? It has two copper ions in its active site. Copper ions are in the +1 oxidation state. Each copper is coordinated by 2 in his residues, and the deoxyform of hemocyanin is colorless. So try to answer these two questions, like which of the following is false regarding hemocyanin, and which of the following is false regarding myoglobin and hemoglobin. Try to find out the answers. Which of them is correct? Maybe you, if you know, that is fine. If not, try to find it. Yes, so for the first one, that is correct. That is, each copper is coordinated by three ends histidine residues. So three C is the incorrect one over here. So hemocyanin has two copper ions in its active site, where all the copper ions are in the plus one oxidation state, and the deoxy form of hemocyanin is indeed colorless, and for each copper is coordinated by three N histidine residues. Now for the next one, what will be the right answer? So for that, it is option B. That is 2,3-BPG decreases the oxygen affinity of hemoglobin. It does not increase it. So the oxygen saturation is higher when there is lower pH for myoglobin. The iron binding sites are similar for both the myoglobin and hemoglobin. So we had studied about that like a few weeks before. Now oxygen saturation is higher at low partial pressure of oxygen for myoglobin. So option B, which says 2,3-BPG increases oxygen affinity of hemoglobin, that is incorrect because it decreases. Okay. The enzyme and its cofactor involved in this conversion. So there is a enzyme and a cofactor that is involved in the conversion from this particular reactant to a complicated structure. So what is that? Hexokinase Mg2 plus, hexokinase porphyrin ring, phosphatase Mg2 plus, dehydrogenase zinc. It is not that complicated. Someone says dehydrogenase and zinc 2 plus. So basically what is happening over here, that you need to actually find out. So what is happening? The first thing that is happening is, or the only thing that is happening over here is that from one of the OH, one hydrogen is removed and there is a phosphate group that is attached over here. So addition of phosphate group, we all know, is done by kinase. So there is a hexokinase activity that is involved over here, and the cofactor that is involved in this conversion is Mg2 plus. So the phosphate group is added by the hexokinase in the presence of the Mg2 plus, that is the cofactor. Next one. So you have to match the enzymes with their functions. Catalase, peroxidase, lubridoxins and ferredoxins. So in column 2 you can see their roles. So see them and find out what will be the correct one over here. So like D. Okay. So let us go one by one. So what does Okay, let's go with the easiest one, that is ferredoxin. So ferredoxin has multiple iron centers. So 4 is B. So it can be either option A or D. So ferredoxin has multiple iron centers. We know peroxidase, also the role of peroxidase, that is oxidization of organic substances by H2O2. So 2 will be like C. Catalase catalyzes the disproportionation of H2O2, and rubridoxin does not have inorganic sulfur. So the correct one over here is D. That is 1A, 2C, 3D and 4B. Okay. Now, which of the following is or are false about azurine? It has two copper ions. It undergoes oxidation reduction between copper 1 and 2. Each copper is coordinated by two N-histidines and two cysteine residues. Oxidized form of azurine receives only one electron. Which of the following is or are false regarding azurine? So azurine is a 8-stranded beta sandwich protein with copper binding sites at its core, where single copper ions are coordinated by the five ligands in a distorted trigonal bipyramidal geometry. Like there are three strong ligands in the form of histidine, cysteines, and another histidine is there, and two weak axial ligands, glycine and methionine. So the structural feature uh, is of a rigid hydrophobic core and a unique tryptophan residue that provides unique spectroscopic properties. So from the structure, you can understand that it has a beta sandwich structure, uh, a single alpha helical insertion over there, and a hydrophobic core. So from there, you can understand that it has one copper ion. Each copper is coordinated by two N-histidines and one cysteine residue. And the copper oxidation reduction happens between 1 and 2, form 1 plus 1 plus 2, and the oxidized form of azurine receives only one electron. Therefore, there is an oxidation between copper 1 and copper 2. So 1 and 3 are the incorrect ones over here. Okay. Now, there is a saturation plot versus the partial pressure of oxygen of hemoglobin at different pH 1, 2 and 3 as shown above. The order of 1, 2 and 3 will be. So this is theta versus partial pressure. So what will be the the order of 1, 2 and 3? So from the slope you can understand that 1 is greater than 2, greater than 3. So the bindings are more, so at lower partial pressure there are more number of like fractions, bound fractions. So at lower partial pressure of oxygen there are more number of bound fractions. So the number of bound fractions will be of like 1 greater than 2 and 3 at lower partial pressure. So next one, there are two clusters given, cluster 1 and cluster 2. So electron transfer takes place from 1 to 2 and N2 binds with cluster 1. There are different options that are given over here. So which of them is correct? So you see in the image it displays like two clusters which appears to be the components of nitrogenase enzyme complex like a FEMO cofactor and the P cluster like that. So uh, in nitrogenase what happens, the electrons are transferred from the iron protein to the P cluster, that is cluster 2, and then from the P cluster to the cluster 1. So the FEMO cofactor is the site of nitrogen reduction. For N2 binding it's the dinitrogen that binds to cluster 1 where it's reduced to ammonia. So the electron transfer takes place from cluster 2 to cluster 1 and the N2 binds with cluster 1. So option B is correct over here. Now the practice problem, so this match the following says you have polymerase, helicase, guidase and ribosome, and what are the roles of these enzymes. So you have to do the match the following. So what is the role of polymerase? So the role of polymerase is it catalyzes phosphodiester bond formation between the nucleotides. What is the next one? It's helicase. So what does helicase do? It separates two annealed nucleic acid strands. Guidase, it catalyzes DNA supercoiling and enables relaxation. Ribosome, it reads and translates mRNA to polypeptide. Okay. So which is the correct option over here? That is one D, or that is option B. So option B is the correct one over here. Any questions? Any questions from this portion? No questions then? Okay then. Thank you very much for joining. We will again meet in the next week. And anyways, I will be sharing the slides and the recording with the NPTEL team. And I will be also uploading it in YouTube. You can find that in my channel. And if anytime you feel that there is a difficulty, maybe you can mail me or ask me in the next class. Okay then. Thank you and bye bye.